Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Abbvie, Apellis, Janssen, MeriaGTx, Novartis and Roche. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
All right, so let's dive in. Let's start the discussion today. I'm going to ask Dr. Larissa Moniz, who's our Director of Research and Mission Programs, to join me now, just to sort of set the stage for our speakers that are coming. Good afternoon, Larissa. How are you?
I am good. It's so exciting to be here, so exciting to see so many of our community members. And just speaking about Cycle for Sight, I actually participated in my first Cycle for Sight in person this year. Went up and down a lot of hills. My hip's still a little bit sore, but it was a really wonderful day. So I hope anybody else who's sort of interested in participating or donating checks it out.
Absolutely. I wish I had that picture of you at the finish line. You did a great job of you guys in the Toronto event. So before we get to our speakers, I was hoping that maybe you could just give us a brief overview of what is gene therapy and how does it work?
Absolutely. So I think gene therapy is a relatively big sort of umbrella term for a lot of really exciting different technologies. So to sort of set the stage, looking at what a gene is. So DNA, it's our genetic material, and DNA is made up of individual units called genes. And each gene is essentially an instruction for the cell. So I sort of like to think about DNA as being like an entire cookbook, and each gene is a separate recipe. And so the genes provide instructions to build or make molecules called proteins, and the proteins are the sort of molecules that do all the work in the cell. So for example, in a photoreceptor cell, there will be a gene that makes special proteins that help photoreceptor cells sense light. So the purpose of gene therapy is to try and change how a cell is acting by putting in a new gene or replacing a faulty gene. So there's a few different types of gene therapy. In sort of the scientific way, people often use very, very different terms for them. We sort of put it under the umbrella of gene therapy. One of the most common ones that people might be familiar with is gene replacement therapy. And that's because the first gene therapy for inherited retinal disease, which is Luxturna, that is a gene replacement therapy. So it's providing a functional copy for a gene that is mutated. There's also other types of techniques called gene editing, and that's where you change a small bit of the gene, for example, trying to cut out a mutation. And then sort of a third type of gene therapy is when you're putting in a whole new gene into a cell that normally wouldn't be there. And so you see this sometimes in optogenetic therapy, when you're putting in a gene to make a cell that is not light sensitive. And Dr. Barakat will also be talking about other uses for this type of gene therapy that are being tested in wet AMD later on in this session.
That's a great way to start. Now, with gene therapy, we've heard a lot about it in the news lately. It's a very exciting time in Canada, especially for gene therapy right now. Can you talk a little bit about that?
Yeah, so we've been really excited in Canada because Luxturna, that gene therapy I mentioned, which is gene replacement therapy for Leber congenital amaurosis or retinitis pigmentosa that are caused by biallelic mutations in specifically the RPE65 gene. So this was approved by Health Canada in 2020, but finally in 2023, it has been approved for public funding. So it's now being delivered in 3 locations in Alberta, Ontario, and Quebec. And patients from other provinces are also traveling to some of these provinces to receive it. So it's been I would say, inspiring to hear from some members of our community who have actually received the gene therapy. I also want to thank if anybody out there signed the petitions and joined us in our advocacy efforts trying to move this to public funding. We really couldn't have done it without you, and I know that the patients who received the therapy are so thankful. And now we really just can't wait to hear what will come next because, Luxturna is just one gene therapy. It was only for maybe about 50 individuals in Canada, and we know that there are so many other uses for gene therapy. So I think I think this will sort of set the stage to start talking about some of these other exciting innovations that are coming up.
Absolutely. That's a great introduction. So I think what we'll do is I'm going to turn it over to you and Dr. Barakat, and then we'll do all of our questions at the end, just so everyone knows. We'll have a common like Q&A at the very end after our speakers. So I will turn it over to you.
Okay, thank you. So yeah, welcome Dr. Barakat. So I'll just give a quick introduction and then sort of jump into the question. So Dr. Barakat, he is the Director of Research at the Retinal Consultants of Arizona, which he joined in 2014. He received training at the University of Pennsylvania and Hahnemann University and has training in both medical and surgical retina. And today, Dr. Barakat, he will be primarily talking about his work, acting as a principal investigator and consultant for REGENXBio. So welcome, Dr. Barakat.
Well, thanks so much for having me. My pleasure.
So at the sort of introduction, I talked a little bit more about inherited retinal diseases, but actually the focus of your part of the talk is wet AMD. And so for anybody in our audience who has wet AMD, they probably know that it is most commonly treated with injections of a type of medication called anti-VEGF. And so the medication is quite effective. And so I was wondering if you could tell us a little bit about why REGENXBio is exploring a gene therapy option for this type of condition.
Oh, no, absolutely. It's a great question. So it kind of goes back to the roots of this question go back to the way you describe different gene therapies, right? So the most intuitive gene therapies, if you have a missing or mutated gene, to replace that. But what REGENXBio is trying to do is trying to essentially have your eye create the medicine that otherwise I would need to inject in the clinic. You're absolutely right. These anti-VEGF injections are very, very effective. They're designed to be somewhere between monthly or every 2 or every 3 months of injections, but they're not a fix. That's the problem. I tell all my patients, this is a Band-Aid solution. It's a wonderful Band-Aid, and it's necessary. And it's really been a huge game changer since it's been out for the last 15, 20 years, but I can't get you off of it. Or it's actually very rare they can get off of it. And so this approach basically means, okay, we realize that we have many patients that need these injections routinely. Coming in once a month, once every 2 months ad nauseum is not the greatest prospect in the world. Wouldn't it be wonderful if you had a treatment, a gene therapy, to use your cookbook analogy, you give the recipe for the medication to the eye. The eye is like a little factory. It starts building it on its own, and you're essentially treating yourself. Now, is it perfect? Will you need supplemental treatments? It's possible, but the idea is, has a lot of potential.
It sounds just so futuristic in a way, doesn't it? Like putting a drug into your eye and your eye producing it for yourself, almost like a self-healing mechanism, which I know that's not what it is, but it's pretty exciting.
Right, no, it's very exciting. I mean, in a perfect world, it'll put me out of business as an injector in the clinic, and that would be wonderful. That's the goal.
Yeah. So I was wondering if you could tell us a little bit about how the clinical trials with the treatment, which is called RGX-314, are going.
So, I mean, they're going quite well. So they actually have a two-pronged way of administering it. So one is surgical and one is in the clinic. The surgical is further along in terms of the research. It's already been past their phase 2 and they're currently in the phase 3 trials. So phase 3 trials are the pivotal trials that are towards the end of when they actually are seeking approval. In the earlier trials, we've had patients that had this surgery. You do a vitrectomy. It's a surgery that's an in-and-out procedure. You vacuum out the vitreous, the floaters, and then you actually inject the subretinal. You inject the RGX-314 underneath the retina. And then essentially what you're trying to do is program the cells of the retina in that area and also the cells underneath that retina called the RPE to produce the medication. We have at this point 3-year, 4-year follow-up of different cohorts, meaning different strengths of this medication, because we're trying to determine which strength is optimal. And in these cohorts, we've actually had some nice results where there's been a drastic reduction in terms of the number of injections needed. So what you'll look at is on an annualized basis, some of these folks only need, say, 2, 3, 4 injections, whereas before they might have needed 8 or 9 injections per year. And somewhere in the range of 50, 60, sometimes 70% of patients can go an entire year without needing an injection. Now, that's the efficacy or early data on the efficacy. We don't have the final word yet. We're still waiting on that. That's still ongoing, actively being recruited in sites all over. But in terms of safety, it's actually been well tolerated. There's always going to be some degree of safety concern whenever you operate on someone. There's always going to be a risk of infection and vision loss, but it's typically very manageable. Otherwise, we wouldn't be doing surgery in the first place. But no, so far it's looking quite promising. That's the surgical delivery route, and then there's also the clinical delivery route, and that's an injection, a little bit different than the typical injections that you get in the office in terms of the anatomy. So instead of injecting it into the vitreous where it floats around where the vitreous is, or the floaters are, if you will, it's a partial thickness injection where it kind of hugs the inside layer of the sclera, the white part of the eye. And there, same exact medication. The whole idea is, can we get the benefit without taking patients to the operating room? And this is earlier on in the trials, but there's some signals of efficacy as well. But you always have to worry about and always have to be careful with possible inflammation, because sometimes these gene therapies can cause inflammation because they're associated with viral vectors.
So yeah, there was a lot there. So I first want to just say hi to Dr. K. Thank you for joining us. So yeah, we're having a conversation with Dr. Barakat, and then we will do a short video of Dr. J. Barth, and then we will get to your interview section. So just sort of sit back and enjoy for the next 20 minutes. Yeah, so what phase are the surgical trials and the clinical trials at right now?
So surgical trials are in phase 3.
Okay.
So there's 2 parallel phase 3 trials ongoing that have recently actually been expanded. So they're pretty far along, and I believe the suprachoroidal injections are in phase 2.
And you also mentioned sort of an intravitreal injection, which if you were getting an anti-VEGF injection would be the sort of injection you were getting, if I'm correct. Is that correct?
Mm-hmm. Correct.
Would there be a way to deliver the gene therapy through an intravitreal injection?
Well, absolutely. I mean, this is not something being explored by REGENXBIO right now, but intravitreal injection in many ways is probably the one that the retinal surgeons are most familiar with. It's most convenient. That's what clinic is set up for. That said, it's also the delivery method that has the reputation for having the highest risk of developing inflammation. And so are there companies that are looking at this right now, and are there companies that are trying to mitigate that risk? Absolutely. But the rationale for going in the suprachoroidal space, it's in a different area of the eye. And the theory, at least the thinking is, will that be somewhat protective? For the same reason, surgical, I mean, let's be honest. Who wants a surgery on their eye if you can get the same benefit from a single injection, right? The reason why the surgical is further along is because we know when we actually inject the medication, the gene therapy underneath the retina, that is a protected, immune protected space. And so it actually reduces the possible side effects of the inflammation.
I think that's so interesting to think about how much of the way the delivery works is really trying to not only get the gene therapy there, but also then to protect from inflammation, which can causes the body to sort of reject the treatment or cause obviously other damage in the eye if there's too much inflammation. So I think that's a really important point. I was wondering if you could also talk about, both from sort of preclinical studies as well as what you're seeing in the clinic, about if there are any sort of long-term side effects from having the anti-VEGF there all the time, as opposed to, if you go get an injection, it goes up and then it goes down again. But if you have VEGF there, it's there continuously. Are there benefits, drawbacks to that type of treatment?
Again, fantastic question to which no one has the full answer, unfortunately. So we know from other trials, for example, the port delivery device, where you actually surgically implant a small device. Think of it almost as if you're diabetic and have an insulin pump that's constantly giving insulin. This is actually a little device the size of a grain of rice that you actually fill with an anti-VEGF. And so it's constant dosing as opposed to monthly dosing. We know from those trials that patients have done quite well. We also think that if there's less fluctuation, meaning less up and down in terms of the fluid or the swelling or the bleeding, what have you, that is more likely to be seen with these dosing as needed, then in the long run, we suspect that's better for your vision. What we don't know is if you have it constantly, if your eye's constantly being treated, will there be side effects? There is a concern. There is a concern that it might be linked to higher rates of atrophy or wasting away of some of the areas of the photoreceptors. That's never really been proven. That's always been a soft sort of signal in prior trials. So from a practical perspective in the clinic, if you take research aside for a second, from the clinic, you say, well, I have to treat the wet macular degeneration, and then we deal with the atrophy, God forbid, if it happens later on. With gene therapy, the other question that's specific to it is, yeah, you're right, you can't turn it off. Right? With a port delivery, you can maybe not refill it. With gene therapy, you cannot turn it off. And the question is, how long does it last? Mm-hmm. It would be, in some ways, both wonderful and terrible to think that it will last forever, right? And that's why these patients all will be in a 5-year-long follow-up. So we have long-term data, and we're still getting that long-term data. But as I mentioned in the earlier trials, from the phase 2 trials for the surgical approach, we now have 3-year and 4-year data of some of the later cohorts. And so far, they've been doing really well.
That's really great. And do you have any sort of input from some of your patients who are, like, have maybe had the regular injections before and now have, sort of a reprieve from it? How do they feel about this?
Some of them just absolutely love it. Some of them absolutely love it. I've had a few that already where it's like, sign me up for the other one. I'm like, okay, slow down. The one consideration that you have to, it's all about setting expectations, right? And so, do I have some patients that have needed a single injection after the surgery? Absolutely. I've also had a few where they do need some touch-up injections every now and again. But at the end of the day, if they've gone from needing it monthly before to every 4 months, that's still quality of life.
Absolutely. We know for some of the work we've done in quality of life studies that there's so much potential fear and anxiety around getting those like very regular injections, and also the impact it has on their family who have to help them go to the appointments and other things like that. So being able to reduce the number of injections somebody gets, I think that's probably everybody's goal. And sort of a final question, just sort of something that maybe, what are you excited about in sort of the broader landscape of wet AMD research or even gene therapy?
Well, I would have said gene therapy, but we already talked about it. I think sticking with the theme of gene therapy is once we, maybe not perfect, but once we have a better handle on the delivery, on controlling the side effects, on knowing how long things last, and on, frankly, is it better to be surgical? Is it better to be in a clinic? Once we have all those things relatively perfected, then there's really very little limit to the potential for this application, because then, as you mentioned before, gene therapy is all about bringing that one recipe back there. And so if you already have figured out how to do that one, you can easily switch to that with something else. So if let's say, for example, a new medication comes out for wet AMD, and there's currently trials for VEGF-A versus C and D blockade. There's current trials for tyrosine kinase inhibitors, and basically all these are medications with the hope of longer duration or perhaps a little bit better efficacy. But once we've proven that these small molecules may have a benefit, then it's only a natural next step to actually put them in gene therapy once we know what we're doing.
Well, I'm excited to hear more as the trials develop. So yeah, thank you, Dr. Barakat. So we're going to move on to our other speakers. So if you want to stay on the line, but if you want to put off your screen or put yourself on mute, and we'll get back to you with questions.
Thanks so much.
Thanks.
Great. Thank you so much, Larissa and Dr. Barakat. So I'm going to introduce our next speaker, who's Dr. Jay Barth. And as I mentioned at the beginning, he wasn't able to join us in person today, but he still wanted to share information about his work with Stargardt. So we have a pre-recorded segment I'm going to play for you now. Dr. Barth, just a little bit about him. He received his MD from the University of Pennsylvania School of Medicine. Dr. Barth has wide-ranging clinical development experience across many therapeutic categories, including gastrointestinal and pulmonary disease, as well as ophthalmology. Dr. Barth is currently the Chief Medical Officer at Ascidian Therapeutics, which is developing medicines using a new approach to gene therapy called RNA exon editing. Ascidian's lead development program is the gene therapy for ABCA4 related retinopathies, including Stargardt disease, and that's what we talked about today. Good afternoon, Dr. Barth, and thank you so much for joining us at Viewpoint today.
My pleasure.
I was hoping we could start talking a little bit about the company. You're the Chief Medical Officer of Ascidian Therapeutics, and you're looking at how rewriting RNA can help treat disease. So could you tell us what the difference is between DNA and RNA, and why at Ascidian you're focusing on RNA editing?
Thanks for inviting me and speaking to you about what we're doing at Ascidian. I think it's a great place to start because knowing the difference between DNA and RNA and understanding what we're doing is pretty fundamental to the approach we have. DNA, I think people know it as the blueprint for the cell. It's really instructions for the cells to make proteins, and proteins is how everything functions in our bodies, including, we'll get into ABCA4 protein, which is important in Stargardt and other diseases. So there's DNA, which is a gene, but there's an intermediate step between DNA and protein, and that's RNA. So the DNA codes for RNA, it's a different molecule, and the RNA codes for a protein. And traditional gene therapies really are focused mostly on DNA, either adding DNA to the cell or changing the DNA that's already in your cell. We are addressing diseases by making changes in RNA. And that avoids some of the concerns, maybe the long-term concerns that people have about making changes in DNA, because those are permanent changes. We have the advantage of kind of having the best of both worlds, because the medicine that we're developing is giving a DNA molecule. So it's just a one-time dose that should have a permanent effect. But inside the cell, what is producing is copies of RNA, and the RNA is the what we call RNA exon editors that replace the RNA that has mutations in it. And that way we have the benefit of the long-lasting effect of a DNA treatment, but what it's doing inside the cell is making copies of RNA, and that's doing the correction of the mutations. So a patient who has a particular mutation could benefit from that type of editing of the DNA. That's as if you had a book with many, many letters in it. Let's say that's your genome, all your DNA, and you change one letter in the book. Well, that'll help the patient whose problem is that that letter's wrong. That's their mutation, but that's very, very specific. What we're doing with RNA is the ability to edit it or replace, we'll call it exons, which are sections, kind of like chapters. So we can replace whole chapters in the book. And that way, anyone, any individual who has a mutation in the DNA, in any letters in any of those chapters, will just replace all those chapters in the book with the normal letters. And that's what we call RNA exon editing. And we feel that has a big advantage as well.
Thank you. That was a really good description of that. I like the book analogy. It really helps with the distinguishing between the RNA and the DNA.
Thanks.
So you're testing this RNA therapy right now with Stargardt disease, which is an inherited retinal disease that affects the macula. Vision loss occurs when there is death in the photoreceptors. And I'm afraid I don't know the number, but I know that there's a huge proportion of Stargardt's cases that are caused by this particular gene mutation in ABCA4. And I was wondering if you could talk a little bit about that mutation and what sort of challenges that might pose for traditional gene therapy, and going into them, why are you looking at RNA editing for this particular gene?
Yeah, as you said, ABCA4 gene, the DNA, if it has a mutation, is the main cause of Stargardt disease. There are some others as well, but this is the primary one. In fact, ABCA4 is associated with other forms of retinopathy, but Stargardt is the most common one. And as I mentioned, there are some limitations to traditional approaches. In gene therapy, also called gene replacement or gene augmentation, the medicine is the gene. It's the DNA that you're giving. So the problem, though, is that certain genes, like ABCA4, are too big to fit into the virus that's used for gene therapy. So the way gene therapy works mostly is that a virus called AAV carries what you want it to carry into the cell, say the DNA, and puts that correct DNA into the cell. But there's a capacity maximum that this AAV can hold. And it just so happens the ABCA4 gene is quite large and too big to fit into this AAV virus. So therefore, that approach, traditional gene therapy, just can't work in this case. Another approach is gene editing. I mentioned CRISPR, and there's other methods that can be used for that. And in that method, you're changing one letter at a time. It's like kind of one letter in the book. Well, that'll help individual people who have Stargardt disease due to that mutation in that particular letter. Just so happens that in ABCA4 retinopathy, Stargardt disease, there are many, many different mutations along that whole gene. Again, it's a big gene that could cause the disease. In fact, more than 900 different mutations have been discovered. So taking a gene editing approach in the DNA, you could fix one mutation at a time, but that'll help a very narrow, number of patients. By taking the approach that we have of replacing these exons or chapters, as it were, of the RNA, one medicine could treat many patients. And the medicine that we have in development right now is intended to replace the first 22 exons of the RNA of the ABCA4 gene, called the first 22 chapters. There are 50 exons in total. So we're addressing 1 to 22. And most people who have Stargardt disease have at least one mutation in exons 1 to 22. So the hope is that it'll treat a large number of patients with different mutations with just one medicine. And that's where we think, this approach has an advantage over traditional gene therapies.
Absolutely. So could you tell us a little bit about where you are with the research right now, how that's going? I believe you're still in a preclinical stage.
That's right. Fortunately, research is going quite well. It's progressed very nicely. The work that the scientists have done at Ascidian is really amazing over the last few years to optimize this approach, because this RNA exon editing is a new way of treating diseases. And the scientists have spent a lot of time doing many different kinds of experiments, both to show that it's effective and that it's a safe approach. So that's all done. A lot of that work is done, as we say, in vitro in the test tube, in the laboratory. And that was done over a long period of time. A lot of people really focused on making this work because said the traditional approaches aren't really the most suitable for Stargardt disease. And we really want to help bring a therapy forward for that condition. So a lot of that in vitro test tube work was done. And then right now we're in the animal testing stage, which is necessary before going into people. And we're kind of in the last stages of a lot of work has already been done on the animal testing side. Some last work needs to be done before we can go get permission from the FDA to move into the clinic. So we're getting much closer to that point in time. And in the not too distant future, hopefully FDA says it's fine. You have all the data, preclinical data, meaning non-human data that you need. And it's okay to go into a clinical trial in human patients. And we'll get the okay to do that in the not too distant future. So we're really excited about the prospect there. And a lot of planning is already underway, for our first clinical trial, and really are hoping to get the engagement of the community of people who are affected by the condition, as well as the physicians that treat patients with the condition, and be able to get the clinical trial started as soon as we're able to.
That sounds fantastic. I guess you don't have like a timeline, right? You have to wait and see how.
Right.
it
There's
goes.
some testing we still are working on, but it's really in the, as I said, not too distant future.
Perfect.
On the near horizon.
That's great. Now, is Ascidian developing therapies using this RNA editing for other conditions?
There are other conditions that we are studying. We haven't said publicly what they are yet, so I'm not at liberty to say what they are. But certainly this type of approach that I was talking about is very amenable to other eye diseases, as well as other diseases in the body, including neurologic diseases, diseases of the muscle, and frankly, any organ, because the methodology that we're using occurs in every cell in the body. So potentially it could treat a wide variety of conditions. But as I said, we're starting with Stargardt disease as the first target condition that we're looking at, because we really feel in terms of treating it with a genetic medicine, right now, we think this is the best prospect and the most near-term prospect. So that's our first condition that we're targeting.
That's great. I think our audience is very happy to hear that. So beyond the work that you're doing with Ascidian, is there other research that you're really excited about in sort of the gene therapy, gene editing space?
I think there's an amazing types of work that are being done across gene editing, gene therapy, again, that's working with DNA medicines, and RNA therapies. I don't want to comment on the prospects, of success for all of those. I think some of those approaches are quite amenable to treating certain diseases of the eye, but they have to meet certain criteria. In our case, for Stargardt's large gene, many different mutations, we really think, that the approach we have is the best one to tackle a condition like that.
And not to put you on the spot, but when I was researching to talk to you today, I was on your website and I thought there was such a cool connection between your name and what you guys are doing. Could you tell us a little bit why the name Ascidian?
Sure. So Ascidian is a sea creature, and a type of sea creature, also known as sea squirt, that utilizes this methodology that we're using as a medicine, but in this sea creature, the Ascidian, it's part of their normal life cycle, that they edit their own RNA to make changes in their, as I said, in the course of their lives. This was discovered a long time ago, and scientists have been aware of this type of biological activity for a long time, and various professors and investigators in universities have been studying it. But it wasn't until the scientists at the company that became Ascidian really focused on it, because what the scientists, is take this normal biologic process that is a big part of the life of Ascidian, is a pretty small part of the life of a human, but then optimize it, kind of tweak it to take this process that normally occurs, but make it into a medicine that'll help people. So that's the origin of the name, and I think it's pretty neat as well. And it just shows, how much we can learn from nature that will hopefully lead to medicines that'll help people.
Yeah, I love that connection to nature. Well, thank you so much for sharing that and for sharing about your work, and we wish you all the best in getting to those clinical trials before too long.
Thank you very much.
Thanks. Okay, well, thank you to Dr. Barth for joining us virtually, remotely, I guess, from earlier this week. And now I think I'm going to turn it back over to Larissa and Dr. K.
Thank you, Morgan. And welcome, Dr. K. Thank you for joining us.
Thank you for having me. Can you hear me? Is my audio okay?
It is perfect. I can hear you perfectly well.
Perfect.
Yep, so I just want to give a quick introduction to Dr. K, and then we'll get into some questions. So Dr. K is a vitreoretinal surgeon at Vitrio Retinal Associates in Gainesville, Florida. She's been there since 2014. And through her training, she's developed a particular interest in inherited retinal diseases and is actively involved in multiple gene therapy trials, including for achromatopsia, RPGR-associated RP, X-linked retinitis pigmentosa, Stargardt disease, and wet AMD. she also has a research interest in optimizing surgical techniques for subretinal delivery. And so today, even though Dr. K has a very wide range of research interests, and maybe we can get into more of those in the question and answer period, the conversation that I'll have will focus largely on Dr. K's work with Atsena, where she is a clinical ophthalmology advisor. So I was just going to sort of jump right in and ask if you could introduce what Atsena Therapeutics is and what its approach is to gene therapy.
Sure. Atsena Therapeutics it's a clinical stage ocular gene therapy company. We are focused currently on inherited retinal disease, and we have one ongoing phase 1/2 trial that I can discuss today. The LCA1 trial, which involves patients with Gucci 2D associated Leber congenital amaurosis type 1, which I provided the 6-month interim update results, and that's what I am at liberty to go through today, since we've released that at the recent ARVO conference. And we'll hopefully be providing our 1-year update later this year for our update for primary outcome results. So at Atsena Therapeutics, I'm a clinical ophthalmology advisor for them. I actually joined them at their inception, which was right at the beginning of the COVID pandemic in March 2020. We all remember that time. And interestingly, what some of the other speakers talked about today, I am involved in the REGENX Bio wet AMD trial as well. So it was fun hearing that talk today with Dr. Barakat and other intravitreal gene therapy trials as well. And today we can also talk a little bit about some of the other programs in the very near pipeline and in the farther pipeline for Atsena Therapeutics.
That is wonderful. So much to talk about. So why don't we start talking about that first trial about the Gucci 2D gene therapy. Can you tell us a little bit both about the condition as well as about the gene therapy and the progress so far?
Right. So LCA1 is an autosomal recessive disease caused by 2 mutations in the Gucci 2D gene. Interestingly, the Gucci 2D gene can also cause a dominant form of LCA, which is or cone rod dystrophy, which is different. And the disease that we're talking about today is the recessive biallelic version of Gucci 2D, which is called LCA1. And this disease interestingly results in very early vision loss, so childhood early vision loss and profound visual loss, but very interestingly and very important for gene therapy, relatively retained retinal structure. So the retinas look good. The OCTs of the retina, optical coherence tomography pictures of the retina look good. So the retinal structure, more importantly, the macular structure is relatively normal. And so we have good structure to be able to go in there and restore the protein that's missing and be able to turn on the function and be able to potentially restore. The mechanism we understand is RETGC1 is a protein that's expressed in photoreceptor outer segments, and that's a key enzyme in photoreceptor. Hold on one second. You guys need to go right back outside. I'm doing my Zoom right now. So thank you. Go right. Live Zoom.
Exactly.
She wants water though. Okay, here you go. You can have that and go right back outside. Thank you. There you go, girls. So, without RETGC1, the photoreceptors get stuck in a light adapted state and they can't reboot. And so they're stuck and patients can't see. They're both they both have visual acuity loss as well as loss of sensitivity and also are night blind as well. But again, they have relatively normal retinal structure. And so what Atsena did, and Shannon Boy and Sanford Boy and the Atsena Therapeutics team have been working on this for years and years and years, but have now gotten this into the clinic with exciting results that I can share today. So ATSN 101 is the product. It's an AAV5 vector. So importantly, it's an adeno-associated viral vector type 5, and it is subretinally delivered, as we heard from Dr. Barakat today, similar to the REGENX Bio trial. So we take patients to the operating room, do a subretinal injection to deliver that RETGC1 protein to photoreceptor cells. And I guess we can skip things on what the promoter are. It's a human rhodopsin promoter. In this trial, in the phase 1/2 that I presented 6 months results at ARVO just a few months ago, we had 15 patients that were treated in this phase 1/2. So it is fully enrolled, and that 15th patient has been treated. We're actually going to be able to give our 1-year results relatively soon. There were 3 different doses, as is commonly done in a gene therapy. So dose escalation in part A with low dose, medium dose, high dose. For those interested, it was 1e to the 10, 3e to the 10, and 1e to the 11 vector genomes per eye. And that high dose was actually chosen as the best dose by the DSMC and by internally within the company to move forward into part B, which is the expansion part of the study, in which we continued to treat 3 more adults and 3 more pediatric patients aged 6 to 18. And so the inclusion criteria, of course, are patients with Gucci2D associated LCA1 and retained outer nuclear structure on their OCT. The primary endpoint of any phase 1/2 is safety. So I'll get to the safety results. And the secondary endpoints were best corrected visual acuity, full field stimulus testing, which is a fancy visual sensitivity measure done in patients with poor fixation. So patients that fixate too poorly or had nystagmus, which is common in this disease, to do perimetry, we can still measure their light sensitivity. We do it with a color dome box of electrophysiology type equipment and flashlights and a big dome, and patients click a button when they see light. And that's called FST. Really important outcome metric that's been extremely useful in some of the Foundation Fighting Blindness studies, consortium studies I'm involved in at showing progression over time. So really useful outcome metric for patients with IRDs. And then mobility testing, which we actually use the Spark Therapeutics. You probably all heard of Luxturna and Veretigene and Parvovec, I should probably say, since that's the trade name. And that used the multi-luminance mobility test. So we use that, we obtained that, licensed that from Spark, and we're able to use the MLMT in the study because that is a historically approvable outcome. So of course we wanted to see what that looked like in this patient population and the visual function questionnaires and other things. The age ranges were 12 to 76. Interestingly, our 76-year-old was one of our better responders. So that's really encouraging. People always ask, is there a certain age cutoff? For this disease, probably not, because the retinal structure is so healthily so retained and so normal. And so again, our 76-year-old was one of our better FST responders. From a safety standpoint, there were no ocular SAEs related to drug. There were ocular SAEs related to surgery, as we see with any subretinal gene therapy surgical trials, that patients all went back to their baseline vision and had an improvement, had resolution of that related SAE. And again, they were surgery related, not IP, not drug related, not related to the LCA1 gene therapy product. And then importantly, and everybody wants to know this, what does the ocular inflammation look like? So we all want to know, what does the inflammation look like? And in this trial, it was very, very good. I have to say, after having seen a lot of different inflammation, a lot of different trials, having been an investigator for 20 or so trials that involved gene therapy, this profile looks very good. And so there were 8 events of ocular inflammation in all of the patients that were related potentially to the drug. They were grade 1 or 2 in severity, and they all resolved with steroids. So either reintroduction of steroid, short pulse reintroduction, and patients were able to get back off of those. So definitely no SAEs related to inflammation, and very, really minimal safety signals, tiny subretinal deposits in one patient, the sort of the typical things that we see in vitritis, a couple cells, trace cells, things like that, things that were very well tolerated. None of the patients discontinued due to any of their AEs. So now regarding efficacy, so BCVA, which was, you would never predict BCVA to be necessarily an approvable outcome in these IRD patients with very low vision. Importantly, nobody went down. So I think that's really a take-home point. There was no loss of visual acuity. These were subretinal injections detaching the fovea and macula in this disease. There were 2 high-dose patients that actually demonstrated BCVA improvement of greater than 0.3 logMAR, which is what our FDA in the US wants. They want those 3 lines of ETDRS improvement. They're kind of stuck on those 3 lines. So we had 2 patients that actually would have kind of met that logMAR improvement, and they were our high-dose patients. FST was really our most powerful dose escalation analysis type informative outcome measure, and showed significant efficacy. So we had significant improvement in FST across all 3 colors tested with dark adapted FST. That's white, blue, and red on the FST testing. And that was specifically the best in our high dose patients. And so when I say it helped inform dose escalation, FST was okay and low and okay and medium, but looked just amazing in our high dose patients, with, some patients even a 20, 30, 40. So a range of improvement in decibels was over 20 in some of our patients, which is really clinically meaningful and clinically significant FST improvements. And that helped us decide to pick that high dose cohort to move forward into dose expansion. The MLMT also, that maze, so we send patients through the Spark MLMT maze. The FDA, for purpose of explanation, they want a 2-step improvement. That's what Spark Luxturna got their improvement with. So that's what what we're going for, is a 2-step improvement. There were 4. So we introduced this mid-trial. We didn't have the license for Spark, and we introduced the MLMT kind of to the mid-end of the trial. And so we were only able to test sort of half of the patients with MLMT. Of course, as we move this forward into a phase 3, we would test all the patients with an MLMT. But 4 of the subjects demonstrated either a maximum MLMT score of 6 or a greater than or equal to 2-step improvement, which is what, again, is that FDA approvable. And this was out of the 5 subjects that were treated, so that were tested. So sorry, only 5 patients had the MLMT. 4 of the 5 showed either that maximum score improvement or a greater than or equal to 2-step improvement. And we kind of learned as well from that if you scored really well at the MLMT at baseline, it had a great ceiling effect. Like if you already scored 6 at baseline, those patients weren't going to have a response because they already were at ceiling, correct? So we learned, like lots of companies, what would be helpful for inclusion and exclusion criteria for MLMT baseline. Patient-reported outcomes also improved. So we had some visual function improvements on VFQ-25. So really exciting. It's fun to actually get up as be able to present positive data for a phase 1/2 gene therapy trial, and that's what I was able to give at ARVO this year.
That was a ton of information. Thank you so much. So just for some of the audience who might not know some of the terminology, so SAE, that is a severe adverse event. And so you were saying that there were not too many severe adverse events, which is exactly what you want to see in a clinical trial. And MLMT is the name of that maze. It's like a multi-luminescent something, mobility.
Luminous mobility test, that's a maze. That's the SPARK maze patients went through for the Spark Therapeutics Luxturna approval. And we, the team, Atsena team, were able to obtain that, license that, and use that MLMT maze, that exact maze, for 5 of the patients. So again, only 5 of the 15 were able to test with that, and 4 of the 5 showed that sort of approvable endpoint. Again, just refocusing that this is really a phase 1/2 safety trial, but I'm really interested in efficacy signals as well. I know patients want to hear about that. And then yes, an SAE is a serious adverse event. We always see them in trials. And so the question is it drug related, or is it surgery related, or is, the patient just had a heart attack because they're 95 years old? So I'm just giving examples. You can have SAEs mean a number of different things. And so there were no drug-related SAEs in this trial. So no drug-related serious adverse events. And then there's always going to be lots of AEs, adverse events. And then the question is, were they inflammation related? Because that's really the gene therapy question, is there causing inflammation? And that was very low, grades of like 1 or 2, and all resolved with steroids, a typical, very minimal steroid reintroduction that we do in these types of trials. So really good.
Yeah, so very, very promising. I know that at Atsena, they're also investigating some gene therapies for other conditions. I was wondering, I don't know what is sort of available to talk about, but I was wondering if you could tell us a little bit about any of those other products in the pipeline.
Yes, so the next 2 on the very immediate pipeline is the X-linked retinoschisis trial. So XLRS is an X-linked condition. We probably have patients on the line today with this disease. So the update on that is the phase 1/2 is about to enroll very, very soon. I can't give maybe exact dates on that, but let's just say very, very, very soon, about to enroll our phase 1/2 X-linked retinoschisis trial. Sites are already chosen and ready to go. And so we very soon, in the very near future, will be starting the X-linked retinoschisis trial here in the US. This is a disease that is predominantly seen in males, given that it's X-linked. And so the RS1 protein is not functioning correctly in these patients, and that retinoschisin 1 protein does a lot of different things, but it helps the retina keep its normal connectivity and keep it sort of attached and normal in structure. But it also helps with the synaptic connections between the photoreceptors and the inner retina and sending the signal to the brain. And so without RS1, the structure looks abnormal and the maculas get schisis, which means splitting of the layer. So if you think of like a sponge, and now fill up your sponge with fluid and stretch it out, that's what these retinas look like. The macula literally looks swollen with little cystic cavities, or we call it schisis, throughout the center retina. And that obviously makes the vision blurry. So the patients are usually symptomatic by age 8 or 9, and they become more and more symptomatic. It's a slowly progressive disease. By age 50 or 60, patients can have developed foveal atrophy or RPE atrophy. I'd say maybe around age 60 would be average. And then at that point, they would have a central atrophic scar. And so our goal would be to go in and treat really, before the time of the development of an atrophic scar, and restore the RS1 protein function and improve vision, improve structure. And that is what we've seen in all of the preclinical data. And so now we're going to be starting the phase 1/2 in humans. Of course, safety will again be the primary outcome, looking at safety. I think the critical thing here is that we will be using a very exciting new novel vector called the spreading SPR. If you look at the Atsena website, it talks about the spreading vector. So AAV.SPR was a vector created by Sanford Boy Atsena team that basically the vector, because of its internal capacities, because of how that capsid works, the vector spreads past the area of the bleb. And so when we talk as surgeons about bleb, we're making little injections subretinally underneath the retinal tissue. And normally a gene therapy only treats the area where a vector touched. With this particular capsid, and we see this in our preclinical testing and specifically in the non-human primate models, the vector spreads almost twice the size. I don't want to give exact numbers here because I'll probably be incorrect, but a significant spread past the margin of the bleb edge, allowing treatment of an area that wasn't detached. And so for X-linked retinoschisis, we don't ideally want to detach that fovea because that inner retina is thin and fragile. And so with this technique, we can inject along the arcades, which are sort of outside in the mid-periphery of the retina, inject a small amount of vector, and then inject another small amount of vector on the other side, sort of again, mid-periphery. And then that vector does its magic, which is spread. And so we don't need to detach it. The vector can treat basically that whole posterior pole without having detached the fovea. And so that vector capsid technology is being used in the X-linked retinoschisis program and will be used in other future programs as well in our pipeline. And that again was developed by Atsena. And so that allows us to do a subretinal delivery for X-linked retinoschisis for the first time. This is the first subretinal trial for X-linked retinoschisis. I don't know if patients are aware, just to recap, there were previous intravitreal trials for X-linked retinoschisis where patients had a one-time shot in the vitreous cavity, which we talked a little bit about today with Dr. Barakat, explaining the difference between modes of delivery. Those trials, specifically the AGTC trial, they terminated after 6 months of enrollment because they did not see efficacy. And that's really because intravitreal transduction is very limited to photoreceptors. And so we want to get, this RS1 protein needs to get to the photoreceptor cell. And at this point, the vectors that have been previously used were not able to do that from an intravitreal approach. Plus, they showed some inflammation in those trials because intravitreal therapy can be more inflammatory. So this is the first subretinal approach to X-linked retinoschisis using a novel vector that spreads in order to safely treat the fovea.
That's really exciting. I was actually just looking up the retinoschisis trials today. And so yeah, it's really exciting to hear about this sort of new approach to also, and I can imagine that would be used for so many other types of gene therapies, a therapy that can treat outside of the bleb if it's successful, to really expand, the area that is getting the gene therapy. So the final question that we're sort of asking, just to wrap it up before we get to audience questions, I know we have a ton of audience questions, is what are sort of other areas of research in gene therapy sort of outside of your work with Atsena that you're really excited about?
Well, I am excited about optimizing delivery. We talked a little bit about intravitreal. Dr. Barakat mentioned suprachoroidal. I think it's an exciting, interesting, novel horizon for us. Intravitreal capsid technology is actually, interestingly, one of the things that Atsena is also working on, optimizing the capsid to be able to, like Dr. Barakat said, if we can find something that works intravitreal, isn't that nice? Isn't that much easier just, to be able to. So optimizing intravitreal therapy, if we can get around all of those hurdles of inflammation and how do we transduce the photoreceptors, I think that's an exciting field. Suprachoroidal, if we can get around the hurdle of how do we actually treat the macula. And there was the Orbit device gyroscope bought out by Novartis cannula that can get into the suprachoroidal space and pop under the macula. I think that's interesting, could be risky, but so ways to sort of, I think delivery is always still a question. We develop these really great capsids and know how to treat these diseases, at least in rescue in animals, but how do we safely deliver it to the human eye? And I think and I didn't talk about the third program that Atsena is working on that's in the preclinical, about to do IND enabling studies very soon, is USH1B. And so I think that's another good example of kind of how to get around difficult hurdles. That's a long gene. We have lots of genes. For instance, the Stargardt gene, like Dr. Barth mentioned today, that ABCA4 gene, he's getting around that by doing the RNA editing. So long genes, how do we get around the problem of it that doesn't fit into an AAV vector? So the myosin 7A USH1B program that Atsena is doing is looking at dual vector therapy. So splitting a vector in half and having it recombine. And the NHP data, and that looks very promising, basically increasing higher than host levels, really the protein restoration with dual vector therapy. So that is not yet in the clinic. That's still in preclinical IND enabling study stages, which, the FDA, rightfully so, has a lot of safety requirements prior to getting anything in the human eye. But that's what I'm very excited about, is because I have patients with H1B that are desperate, and I'm watching them slowly lose vision over time. And it's exciting being able to watch these companies develop these therapies and get them ready to help some of my patients in my clinics.
Well, thank you so much, Dr. K. That was such an interesting look at so many different types of gene therapies for IRDs. So I'm going to turn it over to Morgan, and I'm sure you're going to ask Dr. Barakat to come back as well. And open it up to audience questions.
Fantastic. Thank you. So much interesting stuff happening at Atsena and Ascidian and with Dr. Barakat. So we have a lot of questions. I've been trying to answer a few of them in the Q&A, and I can say we're not probably going to get to everything today, but I'm going to try to combine some of the questions together. I have noticed some common themes. So first off the bat, Camille and Patricia and a few other people have asked that they've had genetic testing done, but their gene wasn't found. Is there any gene therapy that could potentially benefit them, or do they have to wait until their gene is found? I'll open that up to the floor if anyone wants to tackle that one.
I can start. So I have a lot of patients in my IRD clinic where we find either a gene that is not currently in a gene therapy trial, or we don't necessarily find any gene. If it's true that no gene has been found, I think one caveat there is let's definitely ensure that it truly is an inherited retinal disease and that we've considered other diagnoses, autoimmune retinopathy. So making sure that your specialist or your IRD specialist or retina specialist has, if genetic testing has shown nothing and there's anything with the diagnosis that's in question, make sure the diagnosis of a true inherited retinal dystrophy has been sort of confirmed as compared to some of the mimicking things. But back to point one, if it's another gene, or if it's just, we still don't have perfect genetic testing on IRDs, then the field of optogenetics is something that I've been really excited about recently. I learned a little bit about nanoscope when I was at ARVO as well, working with, or just meeting with the nanoscope team. Was pretty impressed with some of their data in the space of RP as well as Stargardt disease. And so that's not genotype specific. And so now we're sort of talking and opening up the door for the question of what are some non-genotype specific therapies, and especially for low vision patients, if you've already lost a lot of vision, is there hope for me? Is there something that could be done? Of course, there's stem cell therapy, but that's a little bit more difficult to get straight into the clinic right now. And there's limited clinical trials with stem cell therapy for patients with RP. But optogenetics is a very exciting field where we basically don't care what gene it is that caused the destruction of the photoreceptors. We only care that there's some inner retina left, ideally bipolar cells for the nanoscope team, because that's what they're targeting with their vector, and they're telling bipolar cells, genetically engineering them to become photosensitive and be able to see light. And a bipolar cell, I learned something myself, actually has the capacity to see in the 2100 range, or 2080 range, so that actually has some meaning, being able to take a hand motion or a light perception patient to 2080 would be life-changing. And so if that, truly shows the safety and efficacy we want it to show to get an approval, and it's not there yet, it's, still in the phases of clinical trials, continuing to move forward, then we might be able to have non-genotype specific visual restoration type therapies to be able to offer patients sort of regardless of what caused the photoreceptor destruction in the first place.
Yeah, and maybe if we've kind of touched on this, but another question that we had over email from Lois. Lois had said, can gene therapy create new retinal cells to replace dead or deactivated cells? So could you clarify about that with gene therapy?
Well, just to clarify, if we're using the word gene therapy and what we mean by augmentation replacement gene therapy, then we are not replacing cells. We are delivering a gene to still existing viable cell bodies. So you do need viable cell bodies. Again, for optogenetics, we're not relying on the photoreceptors because we're assuming some of them are damaged beyond repair. We're relying on a bipolar cell. So there still are cells that we're targeting. What it sounds like that email is sort of getting at would be stem cell therapy. And that there were a couple we mean different things by stem cell therapy. We're not typically just injecting a stem cell, where we're typically, so like J-Site was intravitreally injecting retinal progenitor cells and then seeing if they could improve visual acuity. And they had some mixed results, and it's, that program has, again, sort of mixed results would be kind of my general take on that. And then Reneuron was the company that subretinally delivered retinal progenitor cells. Again, some mixed results there, but that's sort of what we think of when we think of some part of stem cell therapy, to actually try to nourish the retina with a retinal progenitor cell. Apellis is a stem cell company that was actually looking at stem cell therapy, what we sort of classically mean more by stem cell derived RPE cells, and delivering that either back in the day with the Lancet publication to patients with AMD or with Stargardt. And then, of course, what are continuing as well with some programs right now. But that would be restoration. So we would need something like a stem cell trial, a retinal progenitor cell, to actually either restore function or restore cell growth.
Thank you so much for clarifying that. A question for Dr. Barakat from Sudesh. So Sudesh is asking about the treatment you spoke of, the REGENXBIO. He's asking if that injection has to go into the eye. He's saying, if DNA is in all of our human cells, why can treatment be administered to another part of the body, or does it have to be in the eye?
No, absolutely. DNA is everywhere. However, we want the treatment effect to be within the eye. So if you had a treatment, a systemic treatment, so basically, if you had a pill that could treat macular degeneration and with few systemic side effects, then you could probably come up with some gene therapy that's not localized to the eye. Although I think that would open up a whole host of issues as well. You always want to kind of localize it and contain it as much as possible. But essentially, what we're trying to do is we're trying to get that medication to be in the eye where it's affecting the cells that you want to affect. And so that's why, for the same reason, you want the cells there to deliver the gene therapy there.
Thank you. And then we have another question here for you from Catherine. So Catherine says, thank you, Dr. Barakat, for the presentation on RxG314. Is REGENXBIO open to adding to their pipeline, specifically looking at AAV-delivered therapies for treating monogenic RDs?
I can't speak to RGX314 or the REGENXBIO. I'm not part of the company, but I know they've looked at a variety of disease states, not just within the eye. And right now, RGX314 is an AAV8 vector, but they're looking at AAV9. They're looking at all. So I would imagine that all things are on the table right now, but I'm not privy to that.
And another question for you from David. How long do you think it's going to be before this gene therapy might be available to the public? And they're asking if it would be available in Canada at the same time. So can you maybe comment a little bit about that process? I can jump in about the Canadian aspect of that question.
Oh, sure. So the short answer is not soon enough. That's always going to be the answer because we have to go through the rigorous process of getting approval. Especially assuming you had a trial with a single patient, which essentially, why would you do that? That's not really a trial. But assuming you had a trial with one patient, you still would have to treat that patient, follow them for at least a year, preferably 2, but at least a year. And so that's year 1. And then you'd have to, it takes about 6 months or so to get the data, look at it, perform statistics on it. Then maybe it takes another 6 months to a year to submit it, in our case, to the FDA. So even if you had a single patient, you're looking probably in phase 3, right? So RGX314 is in phase 3s right now. So it's done all the other phases already. Then you're probably looking at 2-3 years, easy. Of course, a single patient study is a waste of time. So you're looking at, we still need to recruit hundreds of more patients to see if it's effective, if it's safe, and before we have the real data. So imagine tacking another few years on that. So ballpark, I don't know, 4 or 5 years before we would get to that point. But that's my best guesstimate. It depends on the speed of recruitment and depends on ultimately what the FDA wants. As as to when the FDA approval, and then when that gets translated over to Canada, I'm going to defer that to you. But ultimately, part of that is also going to be the strategy, the commercial strategy of the company when they seek approval.
Yeah, and typically we see, if we think about Luxturna, for example, it was approved in the United States by the FDA much before it was in Canada. It went through the European Union, then came to Canada, and then there's, again, it's a regulatory process within Canada. It has to be approved by Health Canada, and then goes to the provinces for provincial funding as well. So yeah, not able to give a clear timeline, but it will be a few, probably, it will be longer than in the United States, for example. It will be approved there typically first before it came here.
Thus, in summary, never soon enough.
Absolutely. Another question for you about gene therapy for AMD. So the attendee is asking, the gene therapy you're working on is for wet AMD, and wondering if there's any work being done for patients with dry AMD.
Oh, absolutely. There's gene therapy for dry AMD. It's actually in many ways a great target for dry AMD and geographic atrophy in particular. Geographic atrophy, of course, is when you have the wasting away of the photoreceptors. It's the advanced version of dry AMD. We have one drug that has FDA approval in the clinic here in the US, and we are anticipating the FDA's decision on another drug in the next month or two. I believe it's in August. But those, again, same problem. I mean, thank goodness we have it. We've never had anything before, but these are still injections once a month, once every 2 months, and with really no effective way of stretching them out or doing them less frequently. We don't even have enough data to do that yet. And so if anything, this is a great target for gene therapy in terms of, well, now that we've validated in one, possibly two drugs, the pathway in terms of slowing down geographic atrophy is another way of getting that same mechanism inside the eye. So yes, there are companies looking at this with intravitreal injections. So the typical injection in the office, a one and done, if you will. There's companies looking at this, and Dr. K already mentioned this with gyroscope, not just with the Orbit device, but also with subretinal surgery. Same idea, making blebs. So there's many different ways of looking at this, but yes, gene therapy definitely has a potential role in this.
Thank you, Dr. Barakat. I'll give you a breather. I can ask some questions, Dr. K. So there's a question here from Stavros that you had talked about tackling deficits in long proteins by splitting them into two vectors. Could you talk a little bit more about that? And I believe you said the work that Atsena is doing is in USH2B, but they're asking, would this also be something applicable eventually to maybe USH2A or other forms?
Yeah, so I do have a lot of patients with USH2A. Am I still on? Oh, I think I'm on mute. Am I on mute?
No, you're good. We can hear you, yeah.
And theoretically, long genes could be amenable to dual vector therapy. The current program in myosin 7A, which is USH1B, is really a great setup for dual vector therapy for a number of reasons, the length of the gene, whatnot, and we can still fit it within an AAV vector. That particular trial, so if you recall, myosin 7A or USH1B had been previously in a trial by Sanofi using a lentivirus vector, which is like an 8 to 10 kilobase capacity vector, so it could fit that USH1B gene in it. And lentivirus is not especially great at photoreceptor tropism and has maybe more inflammatory quality than AAV for a number of different reasons, mostly that it was lentivirus, that subretinal trial from Sanofi, although they are still reporting some of their long-term safety data and following their patients, did not really show an efficacy signal, and that was 5 or 6 years ago now. But yes, to answer your question, multiple other long genes could be dealt with dual vector therapy. There are different mechanisms for doing dual vector therapy, which get kind of complicated, something called a hybrid vector or something called a classic dual vector. And so, that's sort of a basic science talk of its own, but the basic concept is sort of thinking of splitting a gene in half and then picking a recombination zone and that those intracellularly recombine and you get the full length protein. And so all of that important preclinical work is establishing and proving and double checking and ensuring that you are indeed making full length protein and what that expression pattern looks like, and then it's restoring function and whatnot. So, but yes, other long genes could be amenable to dual vector therapy.
So interesting. Several people have asked about Luxturna, and sort of to what extent does having Luxturna approved accelerate the development of other gene therapies? So, for example, they're asking about the viral vector. Can the same viral vector, the same processes be used in the development of other gene therapies to help accelerate that process?
So it's interesting. Now it's 2023, and our Luxturna approval was in December of 2017. So we're 6 years in and nothing yet in the retinal gene therapy field. So I don't think it accelerated it as much as we wanted it to, or maybe it should have. And it did not make it an easy shotgun approach of what you're asking with, can we just say sort of, let's get the FDA approved process of AAV vector gene therapy or approved and everything else kind of goes through more quickly. That really is not the case. I've had conversations with Wiley Chambers and the FDA about this exact topic. Basically, everything still needs to show that the same safety and efficacy in a subset of patients that have that disease at the current time. I can't predict what will happen in 10 years or even 5 years or even tomorrow, but currently for right now for recessive disease, we still need to, if we're doing gene therapy replacement, we still need to study a patient population who has that disease and look at the vector in that patient population. And there are reasons why, it doesn't mean just because Luxturna worked, it doesn't mean that a different vector will work in a different disease. There's so many different things at play, and inflammation looks different with different capsids, and we use different promoters depending on the disease. So all bets are off as far as that it'll be exactly the identical story with different therapies. But I think the LCA1 story is actually quite similar to the Luxturna story with some of the improvements. I think LCA1 and LCA2 are very similar diseases. I think we're seeing FFT improvement and MLMT improvement, just like Luxturna, and very similar levels. And so hoping and knocking on wood that, everything continues to look good and hopefully moves into a phase 3. And, that might be our next FDA approved product. Because, I think that, maybe similar forms of LCA with similar outcome metrics. Again, that MLMT, now we have a historical. It certainly doesn't hurt having a historical endpoint that recently got FDA approved. As far as, awareness of a path forward, for an approval for a product.
I always find it very interesting, and I'm going to hopefully not misquote whoever this is from in my brain, but I remember speaking to someone about Luxturna and RPE65 and why that was chosen, and I think the answer they gave me was low-hanging fruit, it's an easier gene than some others to actually work with. So I'm always very curious as to which mutations are being chosen for these therapies and how that process happens. Why choose LCA1? Why choose USH2B? Can you comment a little bit on that?
Yeah, it's an interesting question. It's a great question. And it doesn't necessarily mean the most prevalent. So that's an interesting thing. It's certainly not the most prevalent disease if you pick LCA2. And LCA1 is probably twice as prevalent, but still, the LCAs are way less prevalent than EYF-associated RP, for example, one of the most common forms of RP. So it has to do with a good animal model. The luxury of having a dog model, diseases like achromatopsia, there's a sheep model and a dog model. So if you have a mouse model, a dog model, you're sort of all set for your preclinical testing. And then the disease itself is important. Obviously, you want to have a defined patient population, so prevalence has to not be 0. You have to have recruitable patients for trials. And then you want to have, ideally, retained retinal structure. That was a really great thing for both LCA2 and LCA1, that the retinas remain relatively healthy. And so we don't have large staphylomas and atrophic colobomatous retinas like we have in some forms of LCA, and terrible amblyopia from the onset, so, and even some historical preserved vision at birth, even just a teeny bit of vision at birth before vision is lost does help against the idea of amblyopia. So there's lots of different things with the low hanging fruit, but it's really the animal models and and of course that you can, get a vector and replace it and prove rescue in the mouse and the animal model. And then the disease has a treatable retina, that structure is maintained and you have healthy retinal viable cells remaining in that disease.
Thank you. Thank you for that. That's so we had another question here, and this is a question actually I hear a fair bit, and this is from Uzma, and it's asking if someone had gene therapy, for example, they're saying 10 years ago, so maybe in one of the clinical trials, would they be able to get a different treatment in the future if something more effective comes along, and how would that work?
Yeah, so in the same eye has not been done. I think that's an important writing down concept. We have not done that yet, where we retreat the same eye with another subretinal injection. There are immune reasons why that could be problematic, and we simply haven't done yet. It's not to say it could not be done, but you might have to pulse the patient with significant steroids, and you might have a significant immune response. We have treated one eye and then treated a second eye at various different intervals. Luxturna did that with their phase 1/2. In the MeriaGTx RPGR trial, we're doing that right now as I enroll and treat these patients as part of the phase 3 protocol. And some of the choroideremia trials did that. So you absolutely can treat one eye and then come back and treat the second eye. And it's sort of, there's different caveats of when you want to do that and whether you have to repulse steroids and whatnot. And then, but I think the immune system is a hurdle, and it's something that we don't understand as well as we think we do. And there can be significant, we created a term in gene therapy called gene therapy associated uveitis. We actually created our own form of uveitis with gene therapies, GTAU. And I've given talks on that at different conferences like ASGCT on how we deal with gene therapy related uveitis. And so I think the immune system is something that needs to be respected, and we need to understand how to deal with it. But redosing in the same eye has not been done, and there could be some problems with that. That's the short answer to that.
Thank you. So we have another question here for Dr. Barakat. This came in by email from Marilyn. So Marilyn says that she is currently getting shots for wet macular degeneration. So that's anti-VEGFs. And she's noticing that her vision is still declining even with these injections. And she's just wondering if there's currently any other new treatments available for AMD, wet AMD, other than injections.
Well, other than injections, really not. I mean, I mentioned the port delivery device, but essentially that just delivers the same medication that you would be getting in the form of an injection as a form of an implant. So really what it boils, and of course there's photodynamic therapy, that's the cold laser that we've had, decades ago, plays a much smaller role now because these injections are more effective. So essentially I would tell you, no, it's these injections. Are there different ones? Of course there's some differences around the edges of some of these injections. So you could, not knowing your case, not impossible to say, but say on average, I have this conversation with my patients all the time, that there's certainly no reason not to switch between agents if you'd like to. There's a newer agent out called faricimab or Vabysmo that is slightly different in terms of that it's an anti-VEGF, but also it goes after a second target as well, an anti-Ang2. So maybe it's the next generation of anti-VEGF. That said, none of these agents have ever shown to be more effective in macular degeneration in terms of vision-wise. They may last a little bit longer, so durability could be key. The one other thing I would throw out there is that it's very rare that someone just has wet AMD. You have wet AMD overlying your dry AMD. You start out with dry, and we're starting to look at this again, this whole concept, because now that we have treatments available, so as mentioned, we have one FDA approved, possibly another coming soon for the atrophic effects of macular degeneration. Perhaps it may be worthwhile thinking about adding that to the therapy as well. It doesn't make it better, but it slows it down. Now, the caveat is these new agents that are available or soon will be, they've not been tested in patients that had wet and now have atrophy. They've been tested in patients that had atrophy and then developed wet and were continued on. So early days, but if your vision's getting worse with wet macular degeneration, it could be just scarring, or it could be underlying dry.
Thank you for that. We get that question a lot, so that's really helpful. I had a question, a follow-up for Dr. K from Arlene, talking about the dual vector trials, and just wondering if you have any sense of how long it might be until there are trials for a dual vector approach.
I mean, so the timelines are always tough, but for the USH1B program specifically, we're in the preclinical testing at this point, and the FDA requires very stringent IND enabling studies. And so you develop when you develop a therapy and you've gone through your preclinical testing, you know things are looking good, then you submit your investigational new drug application to the FDA, and you do an IND enabling study. So very specific tox studies looking at what the FDA wants you to look at safety before you go into the clinic. And that actually is a relatively short time of doing those IND enabling studies as you submit your IND, and then you get hopefully the green flag from the FDA to go ahead and go into the clinic. So anyway, it's hard to give an exact timeline, but when a company is sort of in IND enabling studies or about to begin IND studies, we're relatively close to getting into the clinic. Others, if you're just sort of talking about dual vector therapy in general, would be farther. If you're talking about something else where you haven't already started or you haven't submitted your IND, then you're farther away. You could be years away. If you're just sort of talking about, let's create a new dual vector for such and such disease, then we've got years and years ahead of planning, working in the preclinical testing, and then submitting an IND.
Great, thank you so much. So I think we're almost at time, unfortunately. So we're gonna have to wrap up for today. I'm just putting into the chats for everyone if I can spell properly, I'm going to put in our health information email address. So I just want to let everyone know that if your questions weren't answered today, if we couldn't get to them, you can reach out to us through that email, and we'll try to help you find what you're looking for. The other thing I wanted to mention, a couple things. I've just noticed in the questions, there's a lot of things coming in about very specific genes that we might not have been talking about today. And so I wanted to say two things. One, on our website, on the FBC website, I'm just going to put that in the chat as well. We have a page that lists a lot of the clinical trials that are going on right now in the IRD space, as well as some for macular degeneration. So you can go on there and take a look. I know if you're looking at clinical trials, and if you've ever been to clinicaltrials.gov, which is like a big has a lot of different information on trials, it can be quite overwhelming. So we've tried to simplify it a little bit on our website, which I've just put in the chat. So please feel free to take a look at that. And of course, we're happy to answer any questions. Also, for those of you out there who have an inherited retinal disease, we do have the FBC patient registry, which you can sign up for. And then if there are clinical trials that come up in Canada for your particular mutation, you will be contacted. And you're also just kind of putting your hand up to say, I have this condition. So it kind of really helps the landscape in Canada, bringing more clinical trials to Canada as well. So definitely encourage you to check out that. And you know what, I can put that in the chat also. And I'll send an email out as well with all of these resources for you within the next couple of days, as well as a recording of this, because I know it's been a lot of information, so you may want to revisit. So yeah, I guess I'll just wrap up by saying thank you so much to Dr. Barakat and Dr. Kay and Dr. Barth for spending their time with us and sharing so much valuable information. It's been really great.